Okay, welcome back everyone to the 2022 Duke Institute for Brain Sciences Distinguished Lecture and Symposium, representing our world in the brain. And next up we have 3 talks from accomplished and creative Duke faculty colleagues coming at the question of brain codes for reality at very different levels of analysis. And I'm sure you will enjoy each talk and the ways that they connect to each other and to our distinguished speakers' work. So I'm going to hand off quickly to our provost, who will welcome everyone and welcome our speakers.
Good afternoon and welcome to the Duke Institute for Brain Sciences Annual Distinguished Lecture and Symposium. I'd really like to thank everyone at the Institute for their efforts in organizing today's events. Special thanks go to Allison Adcock, Interim Director of DIBS, the members of the Faculty Steering Committee, and also to Colleen Bauer for managing the logistics of the event. I hope you enjoyed learning about the exciting work of the students and trainees at the poster session. The theme of this year's symposium is Representing Our World in the Brain. A cardinal goal for neuroscience has been to understand how humans and other animals represent what they perceive, what they feel, and what they learn within the brain. As methods in neuroscience have advanced over the decades, models of neural representations have become increasingly detailed, both providing insights about the contents of the mind and suggesting new avenues for interventions to address clinical dysfunctions. 3 Duke faculty members, Jennifer Groh, Roberto Cabeza, and Lindsay Lichfeld, will share their work on this topic. Their presentations will illustrate how different areas of neuroscience approach the problem of representation, with broad implications for understanding human brain function and even human nature. We also have the pleasure of welcoming Dr. Marlene Berman from Carnegie Mellon University as our keynote speaker today. Dr. Berman's research explores the basic processes of how the human brain represents our complex visual world and how those processes can go awry in clinical disorders. I thank you for attending this special event today and will now turn things over to Mark Summer, who will moderate the next portion of the event. Thank you.
Great. Thank you, Sally. So we're going to have 20-minute talks here, and please feel free to enter your questions, but we're going to get to all the questions in a session after the talks. So our first speaker is Jennifer Groh. Dr. Groh is a professor of neurobiology, psychology and neuroscience, computer science, and biomedical engineering, and also a core member of the Center for Cognitive Neuroscience at Duke. Jenny is the author of a popular science book entitled Making Space: How the Brain Knows Where Things Are. Her research concerns spatial representations and the neural computations that underlie our ability to connect what we see with what we hear. Jenny's research has been featured in the Atlantic, BBC, and other popular science venues around the world. I've known Jenny for over 30 years, since we were graduate students in the same field at the same time, and her work has amazed me at every stage of our careers. Today, she asks the question, how do we see more than one thing at a time? Insights into how neural representations accomplish the seemingly impossible. Jenny?
Thanks, Mark. So I'm coming to you live from a hotel room in Portugal. And since I didn't want to count on the Wi-Fi being up to the task, I pre-recorded the video of my talk, which Colleen can launch now. Thank you.
Hello. Today I'm going to talk about a really simple and obvious problem, but I think it's one that's been overlooked in recent years. And I'm going to propose a potential solution to this problem and share with you the evidence in support of this idea. The problem is, how do we see and hear more than one thing at a time? The work that I'm going to tell you about today is done in collaboration with Shurja Tokdar, another DIBS investigator in the statistics department here at Duke. Much of the work I'm going to tell you about involves data collected in Marlene Cohen's lab. There will also be some data sets from Vinrik Frywald, Liz Romansky, and Michelle Basso. And the work that I'm especially going to tell you about was conducted by Nayan Jun when she did a rotation in my lab. I'll also tell you about work done by Meredith Schmale, a PhD student in the lab, as well as work done by Justine Griego, Justine Shi, and Sarah Gannon. If I convince you of nothing else today, one thing I want to convince you of is that coding of multiple stimuli is not a solved problem. So in our natural environment, we're constantly exposed to multiple stimuli at the same time, but most work in neuroscience has focused on characterizing the activity of neurons in response to single stimuli. So I think most people assume that it's trivial to generalize from what we know about one stimulus to what we know about multiple stimuli, but I don't think that's the case. There are really two key problems. One is that receptive fields are just simply too big. In V1, foveal receptive fields are about a quarter to a half a degree in diameter. The letter E is probably half of that size, depending on your screen and your distance of viewing, the distance from which you're reading the text. So there can be more than one stimulus within a receptive field. In mammalian hearing, receptive fields don't even always exist. Spatial auditory receptive fields are only found in birds, such as barn owls, and are not found in primates and other mammals. So in monkeys, we don't see evidence for circumscribed receptive fields in which neurons are responding only to a sound at a particular location in space. Instead, we see the kind of pattern that is illustrated on the right here, where activity can vary monotonically as a function of the horizontal aspect of sound location, and the maximum responses occur when sounds are located either all the way to the right or all the way to the left, and there are graded responses sweeping out intermediate locations. So current approaches to what happens when there's more than one stimulus to be encoded generally focus on how to, behave in a sensible fashion that nevertheless loses information. So, for example, in the case of attention paradigms, the focus concerns how does the brain prioritize processing of one stimulus at the expense of the others. Another approach concerns investigating ways in which the brain might normalize signals related to the two stimuli, in other words, compute some kind of an average response. That's not a bad failure mode, but it is a failure mode nevertheless. It doesn't preserve information about both stimuli. So we're interested in how the brain might preserve information about multiple stimuli. So to just recap, our basic problems are that with big receptive fields, there's going to be overlap in the either potential or actual population of neurons that are activated by the individual stimuli. And in the case of rate coding, the problem is that neurons can't have more than one firing rate. So if firing rate is supposed to tell you something about the outside world, you would seem to be stuck. So to just depict that here with a graph, if the neuron is firing at that level, you can infer that there's a sound at that location. If the neuron is firing at a different level, you would infer that there was a sound at a different location, but the neuron can't fire at both of those levels at the same time. So what I want to propose is multiplexing as a possible solution. And this idea is actually a very simple one. It is that perhaps individual neurons actually respond to only one stimulus at a time, and that perhaps they then fluctuate across time, and here I'm going to wave my hands, at some time scale and through some unknown mechanism to preserve information about both stimuli. So first I want to show you evidence from the Inferior Colliculus. And this involves recordings that were initially conducted by Valeria Caruso and were published a few years ago in Nature Communications. So the thing to know about the inferior colliculus is that it's a really early subcortical auditory structure. You can think of it as being roughly equivalent in position to the retinal ganglion cells. It's one of the areas where it is known that auditory spatial signals are rate coding the horizontal angle of the sound. And it is also a bottleneck of information processing. All signals that are going to eventually get to auditory cortex, for the most part, travel through the inferior colliculus. So if the inferior colliculus doesn't retain information about two sounds, then it would seem impossible for the animal to be able to perceive them as well. So in our study, we presented either one or two simultaneous sounds at different frequencies and locations. The monkeys were performing an all the sounds saccade task. They had to saccade to each stimulus that was presented in any order. And we did this simple thing first, and that is we counted spikes in a fairly long window of time, such as 500 milliseconds. And we counted the number of spikes occurring in that time window on each trial. So first, let me show you an example neuron. Here on the y-axis is the proportion of trials that exhibited a given spike count. So the blue trace indicates the spike count distribution across trials when the sound was presented at 1,000 hertz and about 12 degrees off to one side. Stronger response for this particular neuron when the sound was at a lower frequency of 420 hertz and off on the other side. The interesting thing is what happens when we presented both of those sounds together. In this case, we got a bimodal distribution of activity patterns with the two peaks of this distribution roughly corresponding to the responses observed on the single stimulus alone trials. So this pattern is consistent with the neuron fluctuating between responding as if only the 1,000 hertz sound was present on one trial and the 420 hertz sound was present on another trial, even though both sounds were presented on all of those trials. So we formally quantify this by benchmarking according to those distributions observed on the single stimulus trials, as shown here for Poisson distributions. And we then screen the data. So if the responses to the A alone or B alone trials are not reasonably Poisson distributed, and if they're not reasonably different from each other, then there is no point in conducting the next steps of the analysis. But if they pass these screens, then we can ask how the responses on the dual stimulus trials relate to those component possibilities. So one possibility is that perhaps when you present two sounds, it will simply either get a larger response, as if the neuron is summing up its inputs, or perhaps you might get a smaller response if one sound is suppressing the response to the other sound. So we call that possibility an outside, and it's akin to summation or suppression. Another possibility is that perhaps the responses will match only one of the two sounds, so we'll call that a single. Third possibility, and that is akin to winner take all. A third possibility is that perhaps the response on the combined trials might be intermediate between the responses observed on either the A alone or B alone trials, so we'll call that an intermediate. And that is like averaging the two inputs, but it could also be the case that our time window might simply be too long to pick up any fluctuating activity. Nevertheless, what we're going to focus on for this talk is the final possibility, that is, if the spike counts are best fit by a mixture of the A and B response distributions. So that is the kind of fluctuating activity pattern that is of interest to us for this hypothesis. So the results from the inferior colliculus are shown here. This is the percent of conditions that were best fit by these four different possibilities: mixture, intermediate, single, and outside. When I say percent of conditions, what I mean is this is the percent of conditions that had a winning model that was at least twice as likely as all other models, and only including those conditions that had passed those initial screens. So I don't want you to think that it's necessarily the case that 40% of all the neurons in the inferior colliculus show this mixture fluctuating property. Rather, it is just over 40% of the conditions that could be tested in this way and that produced a clean answer. So nevertheless, we're going to focus now on these mixtures and how they compare across other brain areas. So the general idea here is that if fluctuating activity is an important factor for how the brain encodes more than one stimulus, then we ought to see this pattern in other brain areas when those brain areas are confronted with more than one stimulus. So, so here's what we find using additional data sets. So here's the inferior colliculus data that I just showed you, just showing the mixtures. Here are results from 6 different studies involving signals in various visual areas. The 2 bars labeled MF and AL are from the face patch system in IT Cortex, and this is data from Vinrik Frywald's lab. It involved either 2 faces or 1 face and a non-face object being presented. And in both of those data sets, we see pretty strong evidence in favor of mixtures being present in the neural representation. The 2 middle bars are from Marlene Cohen's lab and involve areas V1 and MT, with in a task involving 2 gratings presented adjacent to one another. And then the final 2 bars are from a published data set from Stefan Schroyer involving area MT. This is a published data set, and we also saw mixtures in this data set. Where we did not see mixtures is in an additional data set from Marlene Cohen's lab, in which the two gradings were presented superimposed on one another. And the interesting thing about that is that when you present two gradings superimposed, they form a single object, a fused object that can be called a plaid. So in short, we're seeing this mixture fluctuating activity across a whole range of different stimulus conditions, provided you are able to distinguish those two stimuli from one another, but not when they fuse to form a single perceptual object. Our next question, I should say, is whether or not information is preserved about both of those stimuli at the population level. So going back to our adjacent grading stimuli in area V1, here are 3 example units showing the kind of mixture response patterns on the dual grading trials. And the basic question is, you know, on a trial when this neuron responds as if only A is present, even though it's a combined A-B trial, you know, what are these other neurons doing? Are they behaving randomly? Are they correlated with the first unit? Or what is happening? These correlation patterns are going to be critical for understanding whether or not both stimuli are preserved in the population response. So I'm going to jump ahead to a depiction of what we think is happening at the population level. So this is real data from one recording session, and there are 13 neurons in this recording session. These recordings were done with a Utah array, so there's up to 100 different electrodes. And as it happens in this session, there's 13 that had pretty good evidence for mixture response patterns. We've color-coded the activity on every trial. There were 18 trials. We've color-coded the activity red if it is most similar to the response evoked by stimulus A, and blue if it is most similar to the response evoked by stimulus B. And so you can see here that across any given row, there are both red and blue squares. That's baked in by the fact that I only am plotting the data from cells that had reasonably strong evidence for this mixture response profile. What's most interesting is that if you look at any given column, that is to say on any individual trial, there are also both red and blue squares on those individual trials. So that indicates that at the population level, there are some cells responding as if only A is present and other cells responding as if only B is present on any given trial. Furthermore, at the overall population level, there is a bias in this representation. There are more red squares than blue squares, but there's also correlation between individual neurons. So cells 1 and 2, for example, are both encoding stimulus A. They're both have red squares up until trial 16 when they both flip to blue. So there's correlation between units 1 and 2. There's also correlation between units 3 and 4. But those different units are anti-correlated with each other, and that anti-correlation helps to ensure that both stimuli appear to be represented in this population. Okay, quickly extending this further to a multisensory case and to the case of a decision-making task. So both in our own lab and in collaboration with Liz Romansky, we see that when your two stimuli consist of a visual stimulus and an auditory stimulus, you also see evidence for this mixture pattern in prefrontal cortex for the Romansky data, in Superior Colliculus for my lab, and in a moment I'm going to tell you about some more data from the Inferior Colliculus for my lab as well, from Meredith Schmel's PhD work. But before I get to that, let me also tell you about a decision task. So this is data from Michelle Basso's lab. And the basic idea here is that if monkeys are performing a task in which a ambiguous glass panel to the left or to the right, there are trials in which there's strong evidence for making a choice to stimulus A, or strong evidence for stimulus B, and then an ambiguous case in which the glass pattern is ambiguous and doesn't give strong evidence in favor of either one. So the monkey has to make a guess. And the prediction would be that in these cases we would see evidence for mixture-like response patterns. And indeed, that is the case. About 85% of the SC neurons in the data set that they shared with us can be classed as having a mixture response pattern. Final bit of data just to tell you from Meredith's PhD thesis is that visual stimuli can bias that mixture auditory representation. It's previously been seen in ferret auditory cortex that when you present two sounds, but you present a visual stimulus that matches one of the two sounds, you can shift the activity in favor of the matched sound. And Meredith sees the same thing in her inferior colliculus recordings, where when a light is paired with a visual stimulus, more of the trials are devoted to responding to that sound than to the sound that does not have a visual stimulus associated with it. So to wrap it up, what I've tried to tell you today is that two simultaneous stimuli can invoke responses across trials that are a mixture of those that are evoked by each stimulus alone, that this pattern is found in many brain areas when the stimuli are distinguishable, whether or not it is two sounds, two visual stimuli, or an auditory and a visual stimulus. This is not seen when stimuli fuse into one object and form a plaid. And we see similar findings in a decision task when there are two equally likely choices under consideration. At the population level, information about each stimulus or each choice appears to be preserved, but can be biased in favor of, for example, a visual stimulus that's paired with a sound. Wrapping up with a couple of big picture thoughts. We often think of variability in neural activity as being noise, but I think it's important for us to consider the possibility that it can be signal. Here we have tried to generalize from, we've tried to make the leap from studies involving one stimulus to studies involving two stimuli. And I think it can be seen here that what we learn from just studying one stimulus at a time doesn't easily generalize to cases of more stimuli. But I think one of the things that is a bit elegant perhaps about this multiplexing theory is that it preserves much about what we know from these from neural representations from single stimulus studies. Specifically, we've been able to preserve the notion that it is a mapping between neural identity and firing rate on one hand to some kind of inference about the stimuli that caused that firing pattern on the other hand. However, the mechanism by which these fluctuations encode the multiple stimuli is as yet unknown, and how they may be decoded is unknown as well. So finally, I want to thank everybody in my lab for all their hard work on this and other projects, and note that we have an opening for a postdoc and for a postbac technician, and please email me if you're interested. Okay, thanks very much.
Thank you, Jenny. Enjoy Portugal.
Thanks. I'm looking forward to coming home.
Stick around for the Q&A session, though, if you can. All right. So next up is Roberto Cabeza. Dr. Roberto Cabeza is a professor of psychology and neuroscience and a core member of the Center for Cognitive Neuroscience at Duke. Roberto investigates the neural mechanisms of memory in young and older adults using behavioral, brain imaging, and brain stimulation techniques. He graduated at the University of Buenos Aires, earned his PhD at the University of Tsukuba in Japan, and did his postdoctoral training at the Rotman Research Institute at the University of Toronto. Roberto has over 200 publications, which have been cited over 48,000 times. He's a member of the governing board of the Cognitive Neuroscience Society and of the advisory board of the Max Planck Institute for Human Development in Berlin. Roberto received the Young Investigator Award from the Cognitive Neuroscience Society and is an elected fellow to the Society of Experimental Psychologists. Today, Roberto will address how we remember our representations of the world. All yours, Roberto.
Yeah, thank you, Mark. So how do we represent the world. So the assumption is that the world is represented as synaptic changes over groups of neurons, cell assemblies in different parts of the brain. And then how do we remember them? So this is a simplified cartoon of that process. The assumption is that one event, such as being here, for example, is a combination of different types of information, like such as visual information here in green, and auditory information in red, and also here in blue, what would be the meanings of the different elements. So the idea is that the one event is not stored in one place in the brain, but rather is distributed over the cortex, close to the regions that originally process each type of information, so that the visual information is stored primarily the visual pathways, such as the ventral visual pathway for object processing. Meanings will be stored in regions that specialize in meanings, such as the anterior temporal lobes or the angular gyros, auditory information in auditory cortex, and so forth. So that the idea is that when during retrieval, when we try to remember something, we are able to activate the only kind of integrated representations that is supposed to be stored in the regions of the hippocampus. We can reactivate that region, and that allows us to reactivate all these different memory traces. And that combined reactivation of these traces is what provides us with the integrated experience of having had a past events. So using fMRI, one can study different aspects of this process. Traditionally, people would use univariate fMRI to compare activity, mean activity in different brain regions, and differences in univariate activity are assumed to reflect differences in processes, so the operations we perform in the information. However, in order to study representations, we have turned, like many other people in the field, to use a multivariate activation pattern analysis that allows to study more of the contents of the information. So the assumption here is that differences in the similarity of activation patterns reflect differences in representations. So there are different ways in which we can compare similarity. The simplest way is, for example, is a simple correlation. For example, between encoding when you are information and retrieval when you are recovering that information while you are trying to remember. So that that type of simple correlation is known as encoding retrieval similarity. Another type of simple correlation is when we study correlations among the encoding items or encoding similarity. So that similarity will tell us how distinctive is the representations of each of the items we encoded. So importantly, we can use these measures, ERS and EES, to predict memory performance, so that whether you are going to remember things that actually happen or true memories, or you may even remember things that did not happen or have a false memories. So the first study I'm going to show you used this paradigm in which participants saw scenes paired with the labels such as barn, and during retrieval they only were presented with the barn, and they had to retrieve the original image that they saw, and they rated the quality of that memory from 1 to 4. We used that memory ratings, but we needed to confirm that those ratings were actually good reflections of actual memory, objective memory. So we tested outside the scanner by presenting different pictures of that images and asked participants to choose the correct one. And we found that actually the ratings in the scanner were predicted very well the correct recognition in this post-scanner test. So there are different ways in which we can calculate ERS. One is to do one-to-one, for example, the barn during encoding and the label barn during retrieval, or we can compare similarity across items. The first one we call item ERS because it's similarity for one item, whereas CRS will be similarity across all the sets of stimuli. And this is, you use it as a baseline because it basically reflects just the similarity between any encoding and retrieval trial. So one of the main findings in this study was identifying a left occipitotemporal cortex region where ERS increase as a function of memory rating. So put another way, so the greater the ERS, the stronger your memory. So importantly, this didn't happen only for item ERS and not for other set ERS, suggesting that this memory is specific to individual images. So this type of scene-specific representations then well supported by OTC, but these are true memories. How about false memories? So Eric investigated this in another study in which he presented not just one, but multiple versions of each object. For example, this kettle was presented in different versions. So that allows to study the familiarity elicited by these pictures, and it tends to lead to false remembering. So during encoding, we looked at the activation brain patterns for these different images, expecting that there will be differences even between the different versions of the kettle. We also look at the hippocampus, which is important because it's supposed to mediate a process known as pattern separation that helps distinguish between highly overlapping representations, such as those between the different exemplars of one object. During retrieval, the subjects had to indicate whether an item was old or was new. And calling old an old item is a true memory. Calling old a new item is a false alarm or simply false memories. So our goal was to use encoding similarity to predict a memory performance. So we identified that left occipitotemporal area that overlap with the one I showed you before, where increased encoding similarity predicted not only true memory, but false memory. This is consistent with evidence that that familiarity elicited by similarity among items helps with saying old to the old item, but also leads us to false alarms. So what can moderate this false alarms effect? And that's the hippocampus, because we found that when the similarity of items was low in the hippocampus, then the false memories were reduced. Okay, so these studies focus on a similarity for an object or a scene as a single thing. However, representations are not simple. They are comprise many different features, such as visual and semantic features. So this cannot be studied just with simple ERS or EAS. So we need a representational similarity analysis. In representation similarity analysis, we start with different models of the stimulus, which by mean I think that we need a matrix setting identifying the similarity between H pairs of objects that we present. Similarity, visual similarity, or semantic similarity. For example, here the basketball is similar to the pumpkin in a visual similarity matrix, but is similar to a tennis racket in a semantic similarity because they are both sports equipment. So basically you can have multiple similarity matrices, and then you can link each of them to the brain to identify where the regions coding for that type of features are located. So then we need an equivalent similarity matrix, but using brain activity, and that is obtained by presenting each of the objects and then correlated the activation patterns in a brain region. And finally, the critical point is the second order correlation, in which we relate the stimulus model to the activation pattern in order to identify brain regions that are seems to be storing those types of representations. So in this study by Simon Davis, we presented objects, and the assumption is that the objects will be processed along the ventral pathway for object processing with posterior occipitotemporal areas, so B1 and B2, I mean early visual cortex, will be processed primarily visually, and as you move anteriorly, you start getting into more conjunctive representations and more semantic representations. And here is a key assumption. When a single object is presented, you don't store a single representation, you store multiple representations. You store visual representations, for example, in posterior areas, but also semantic representations in more anterior areas. So we were interested in how these different types of representations predict different types of memory. In particular, we compared two types of memory tests: a conceptual memory test in which participants had to remember the concepts they investigated regardless of the picture, and a perceptual memory test in which they had to remember the exact picture because they had to reject very similar pictures or distractors. So our assumption is that performance in the perceptual memory test will be determined primarily by visual representations, whereas perceptual in posterior occipitotemporal areas, whereas performance in the conceptual memory test will be determined primarily by semantic representations in more anterior brain areas. So consistent with this hypothesis, we found that early visual cortex stored representations that predicted perceptual memory. However, there were also visual representations in the hippocampus, for example, that predicted more general memory. So predicted not only perceptual, but also conceptual memory. As for semantic representations, consistent with the hypothesis, we found that more anterior areas such as perirhinal cortex, which is strongly associated with semantic memory, semantic representations there predicted conceptual memory. At the same time, semantic representations in more posterior areas, such as the angular gyros, predicted also perceptual memory or more general memory. So an interesting question is, how can the same type of representation predict different forms of memory depending on the region in which it is stored? And two hypotheses we entertain is, one is that the nature of representation changes slightly depending on the storage location, because each region has different wiring and different computational capabilities, and that could alter the type of representation being stored. Another possibility is that the effects of representations on memory depends on how the representations interact with memory networks, particularly during retrieval. So it's not enough to lay down a representation, it has to interact with these networks in order to be retrieved. So memory representations could differ in memory impaired populations. I mean, all these studies so far were focused on young adults. So turning to this type of memory impaired populations, study 4 investigated the effects of healthy aging. So 2 points that you need to know about healthy aging for this study. One is that aging impairs most cognitive abilities, but typically not semantic knowledge, as you can see in this classic figure. So whereas most cognitive abilities, including perception, processing speed and working memory, decline with aging. Episodic memory, knowledge, I mean, vocabulary of language, and we know also general knowledge of the world is well maintained during the lifespan, and even, I mean, that helps us that we can keep effective in our profession even where we age. So another point is that aging is associated with a reduction in the specificity of visual representations known as dedifferentiation. So in older adults, brain regions specific to one kind of stimulus, such as faces, respond also to other stimulus, as you can see here. The fusiform face area clearly distinguishes faces from other objects in young adults, but in the older adults, these distinctions are getting lost. So the dedifferentiation has been linked to impaired cognition, and this we investigate in the next study. So most previous studies focus on univariate activity, And so we use a stimulus with that and a stimulus that didn't have much semantic content, such as unfamiliar faces, but we were interested in how older adults present both visual and semantic information and use meaningful objects as a stimuli. So the paradigm is the same that you saw before for study 1. We use both RSA and ERS method, and we focus on 2 regions of interest: visual cortex as the prototype of the visual storing area, and anterior temporal lobes as the other end of the visual semantic continuum. So consistent with the differentiation, we found that the fit between the visual representation and the brain was reduced in visual cortex. So that second order correlation I mentioned before was attenuated, suggesting that the visual cortex doesn't represent as well visual details as in older young adults. Conversely, we found that in the anterior temporal lobes, we actually found the reverse. The semantic representations were actually stronger in older adults than in younger adults. So whereas the first finding is consistent with the differentiation, we found, I mean, this is the first report of this, we found that older adults were showing hyper-differentiation, suggesting that the semantic representations were actually strengthened. And this could be also, given that we know that semantic memory can help older adults cope with deficits in other functions, this can be seen as a compensatory change. Finally, we look at ERS. So an ERS, we found again that 2 regions, early visual cortex and also the hippocampus, we watch another ROI in this case, showed a reduction in ERS consistent with the deficit in memory representations. But again, the anterior temporal lobe showed this age-related increase. Importantly, this ATL effect was correlated with performance in older adults, suggesting that it's actually helpful for performance to recruit ATL in this way. So the finding of increased ERLs in older adults correlated with memory performance, such as also a compensatory mechanisms. So in sum, to conclude, study 1 showed that our memory for an event depends on reactivating during retrieval a representation that is similar to the encoding representation, I mean, of a specific with item ERS, which is what we found in the left occipitotemporal cortex. In study 2, we found that greater similarity across items or EES in left occipitotemporal cortex representations lead to both true and false memories, but false memories are attenuated when representation similarity in the hippocampus is reduced through pattern separation. In study 3, we found that visual representations stored in visual cortex tend to predict perceptual memory, whereas semantic representations stored in anterior brain regions tend to predict conceptual memory. But more generally, we found that the way in which memory representations predict memory depends not only on the type of representations, but also on where they are stored. And finally, during encoding, older adults showed the differentiation in visual representations in visual cortex, but hyper-differentiations of semantic representations in ATL. And during retrieval, they displayed impaired representations in visual cortex and hippocampus, but enhanced representations in ATL. Both findings are consistent with compensation. So taken together, the approach on the representations complement the work on using univariate activity, because the assumption is that processes operate on representations, that the type of the information being held by the brain. So you need both the processes and the representations. Moreover, this needs to be supplemented by network analysis that tells you how these regions interact in order to support behavior. Thank you very much.
Great, thank you, Roberto. We're a couple of minutes ahead of time, which is wonderful. Colleen, do you know, is the Q&A feature activated yet? Can audience members? Okay.
Yeah, people should be able to go ahead and ask the questions there.
Great. I don't see any questions yet. I would just encourage the audience, if you have questions as you're watching these talks, You can get a jump on things by entering them into the Q&A little format there, and then we can be efficient after all these are talks conclude. Okay, so yeah, there we go. So next up is Lindsay Lichfeld. Dr. Lindsay Lichfeld is an associate professor of neurobiology at Duke. She earned her PhD in Massimo Scanciani's lab at UCSD, investigating the role of inhibitory circuits in the hippocampus. As a postdoc at Harvard Medical School, Lindsay worked with Clay Reed using optical approaches to interrogate visual circuits, and also with John Mansell applying behavioral methods to study visual perception in mice. Here at Duke, Lindsay has integrated all of these techniques to study the function of higher visual areas of mouse cortex using neurophysiology and robust behavioral paradigms. Her work is leading the way in advancing our knowledge about the links between neural activity and perception. In recognition of her contributions, Lindsay has been honored with the Director's New Innovator Award and fellowships from the Whitehall Foundation, the Alfred P. Sloan Foundation, and the Pew Biomedical Trusts. Today, Lindsay will talk about sensory and cognitive roles of visual cortical areas. Lindsay?
Thanks so much, Mark. And thanks to DIBS for including me and the mouse visual system in this discussion about how the world is represented in the brain. This is a question that's very close to my interests. And what we're really interested in is understanding what the anatomical and physiological mechanisms are that enable information in the external world to be represented in populations of neurons in the brain, and then how that information is used actually to guide behavior. So one of the most prolifically studied representations in the visual cortex is the representation of orientation. And so this was first observed by Hubel and Wiesel in their recordings from cat primary visual cortex. And what they found was that when they presented oriented bars of light, that they could evoke robust trains of action potentials in neurons in the primary visual cortex. But by changing the orientation of this bar even slightly, they evoked many fewer action potentials, or even none at all. And this was really exciting because it provided one of the first really concrete examples of how a representation in the brain might reflect something very specific about the external world. And since then, in every mammalian species that people have looked at in the cortex, this representation of orientation has been found. In humans, monkeys, cats, rats, mice, even most recently in the armadillo, suggesting that this ubiquitous representation is actually probably really important, not only for our perception of orientation. So not only is this representation ubiquitous across species, it's also ubiquitous across cortical areas. And this is true both in the mouse, as I will show you, as well as commonly studied in primates. So in the mouse visual cortex, as shown here in a tangential section, looks a lot like, in some ways, the primate visual cortex, where you have a primary visual cortex, or V1, an array of different higher visual areas. Whereas in primates, these higher visual areas are known as V2, V3, V4, so forth. In the mouse, they're called by these names like LM for lateromedial, AL for anterolateral, and PM for posteromedial, and refer to their location in cortical space. And so as you can see from this experiment here, what Wang and Burkhalter found is when they injected dyes of different colors into different retinotopic positions in primary visual cortex, they found that these neurons in V1 projected directly to this array of different higher visual areas. And importantly, that these projections maintained their retinotopic organization, suggesting that each of these areas could represent a complete picture of the external world, and therefore might have unique and specialized functions for perception and behavior. And indeed, there's been studies that have shown that neurons in this area AL and PM have really distinct representations for speed, and therefore might be important for representing self-motion versus the motion of external objects. However, today I want to focus on this representation of orientation, which I think is equally interesting because it's actually very similarly represented across these areas. As you can see in this data from Ed Calloway's lab, where when they recorded from neurons in primary visual cortex and then 6 different higher visual areas and measured the orientation selectivity of neurons in these areas, that they found that there was highly orientation selective neurons across all regions. And that while there might be some slight differences in exactly how orientation selective the different regions are, they are all carried this really important signal that could be used to guide perception of orientation. But this brings up this really important question then of which of these areas might actually be used for discrimination of orientation. And so I want to talk, bring this to some really specific potential hypotheses for how this might work. So one possibility is that primary visual cortex, or V1, is sufficient for discrimination of orientation. We know that neurons in V1 make specific projections to downstream motor-related or decision-related areas like the superior colliculus, the striatum, motor cortex. And we know that neurons in V1 have sufficient information in order to discriminate the orientations that are presented. And so it's possible that the circuitry bypasses all of these different higher visual areas and instead directly targets this decision-making process. Alternatively, it's possible that neurons in V1 might project, for instance, to area LM, which is a V2-like area, and that carries the signal to downstream decision-making areas. Alternatively, V1 might diverge onto multiple higher visual areas, although avoiding others, and that could be responsible for the decision-making process. And yet another possibility is that the output of V1 is fully divergent across all of these different areas that encode orientation selective information, and that they're all important for the decision-making process. And notably, part of these hypotheses also could be that this divergence might lead to redundancy, such that no one area is necessary. Or it may be such that the convergence of this information on the decision-making output area is actually the important integration step, such that each of these areas are necessary. So there's lots of possible different circuit proposals for how information might get from V1 to the decision-making process, as well as what the kinds of computations that are going to mediate this. And this is the kind of question that we're really interested in using the mouse visual cortex because of our access to be able to specifically manipulate populations of neurons in these different areas. And so the way that we're going to be able to do this is by using an approach called optogenetics. And so what this is that we can express light-gated ion channels that we then, when we shine light on them, they open, and that allows cations to enter the cell and depolarize the neuron, and allowing you therefore to have rapid and transient influence on the excitability of the cells that express this channel. And so importantly, this channel rhodopsin is polarizing the cells, and so it's making the cells more active. But what we really want is to be able to silence neurons to understand what their contribution is when those neurons, their activity is taken away. And so we can use a trick of a sign inversion in which instead of putting these channel rhodopsin molecules into excitatory cells, we can put them into inhibitory interneurons. And that way, when we turn on the light, we then activate these inhibitory interneurons and thereby robustly silencing neighboring pyramidal cells to test the role in these kinds of behaviors. And so through the combination of targeted light and targeted viral expression, we can then go around and silence each of these different areas and test the effect on the animal's ability to discriminate orientation. So finally, the last piece of this is that what we need is an orientation discrimination task. And so here I'm going to show you a task that we've taught the mice to perform in which they're going to do a go, no go orientation discrimination detection task. And so how this works is that they have a little lever that they use to make their decisions. They place their paw on the lever and they push that lever down to initiate a trial. Then when the trial is initiated, visual stimuli begin appearing. There's a series of vertical gratings, and the mouse's job is to detect a change in the orientation of those gratings. And in particular, when the orientation changes, the mouse has to release the lever. And if it does so within a short reaction time window, it gets a juice reward. Alternatively, if it releases the lever too early or too late, then it gets a timeout. So here's a movie of the mouse performing this task. So he pushes the lever down, you see the visual stimulus appearing. And then after that orientation change, the mouse released the lever and he got his juice reward. Here's another trial. And on that trial, the mouse released the lever following a distractor stimulus, and we call that a false alarm. So we can now use this behavioral data to assess whether how suppressing different cortical areas influences the animal's hit rate and its false alarm rate. And so again, here's a schematic of the task. The animal presses the lever down. This initiates the presentation of these brief vertical gratings. After some variable interval and number of presentations, the orientation changes, there's a target, and the mouse has to release the lever in order to get its reward. And then on randomly interleaved trials, we can activate the light to therefore activate these inhibitory neurons and locally suppress activity within the different cortical areas to see how this affects the behavior. And importantly, since this is an ion channel, this is very rapid onset and very rapid offset. And so we can measure these effects on a trial by trial basis. So here's data from an example mouse. On the x-axis is the size of the orientation change relative to the distractor. So when that orientation change is large, for instance, this 90-degree condition, the animals have a very high hit rate. But as that orientation change gets smaller and smaller and closer to the distractor, the animal's hit rate falls off. And we can then measure the animal's detection threshold. Then both on control trials here, as well as on the randomly interleaved trials where we activate the LED in order to suppress primary visual cortex in this case. Notably, we are tuning the intensity of that light to have a moderate effect on the animal's behavior, such that we're not having a big effect on the animal for the easy trials, but having a relatively a measurable effect during the at threshold. So in addition to measuring the effect on the animal's threshold, we can also measure the effect on false alarm rate. And so this is by determining what the probability is that the animal released the lever following one of these distractors using the same window that we use to identify hits. And so here you can see the animal's false alarm rate was significantly suppressed on the conditions when we turn V1 off. So this is one mouse. Here is data from 6 mice in which we suppress primary visual cortex when the visual stimulus was aligned with the representations of the population of neurons that we're suppressing. And you can see that in all 6 of these mice, we get this robust increase in the animal's threshold, suggesting that V1 is important for the animal to perform this task. We also see this robust decrease in this animal's false alarm rate. So not only is the animal less likely to think to see a change, it's also less likely to think that it saw a change, which is, I think, which is an interesting phenomenon that we can potentially discuss later. So not only do we see this impairment in the animal's behavior when we suppress V1, we also see a similar impairment when we suppress LM and AL, where again, we see this robust increase in threshold and decrease in false alarm rate. But interestingly, when we suppress neurons in PM, we see no effect on either the threshold or on the false alarm rate. So one important control that we need to think about here is whether this effect that we're seeing is really due to the effect on sensory processing, or might be due to some global, more cognitive effect, where, for instance, we're just making the animal less likely to release the lever. And so in order to test for this specific contribution of sensory processing, we performed a control where we move the visual stimulus outside of the receptive fields that we're actually inhibiting. And you can see that in these conditions that these same mice are no longer have an increase in threshold and no longer have a decrease in false alarm rate. So just to summarize what I've shown you so far is that suppression of V1, LM, and AL are all sufficient to impair orientation discrimination, suggesting that each of these areas are in fact involved in the perception of orientation. And that notably that this is only true when the suppression is aligned with the visual stimulus, such that we think that this is a consequence of sensory processing. And this is a really important result, that even though the information is present in V1 and potentially the downstream connectivity is present in V1, that V1 is not actually sufficient for mediating the process, and that we do need activity in these higher visual areas. The other interesting result so far was that PM had no role. And this is despite the fact that many neurons in PM encode orientation and could also potentially be sufficient for decoding the presented stimulus. So I think this is, again, an important thing, that just because you see a representation is present doesn't necessarily mean that it's actually going to be used. So the next thing that we wanted to know was whether PM might actually have some type of role in perceptual decision-making. And so we've decided to interrogate a different task. And this task is a contrast discrimination or detection task. And so the reason that we chose this task is because these neurons in PM also respond robustly to the presentation of contrast. And we thought that this representation might also just be more generally encoded across the brain, and that the brain might take advantage of the activity across all areas. So the organization of this task is very similar. Again, the animal pushes down the lever to initiate the trial. But now, rather than having distractor presentations, the screen maintains mean luminance until a target appears, at which point the mouse needs to release the lever to say that it detected it. And again, we can suppress the cortex on randomly interleaved trials from when the animal pushes down the lever until when it releases it. So here is data from an example mouse. On the x-axis now is contrast. When the target contrast is high, the animal has a very high hit rate that falls off as that contrast gets towards the mean luminance. Again, we can measure the animal's threshold, both in control trials and when we're suppressing, in this case, primary visual cortex. And again, in this example animal, you see this nice increase in threshold. Again, we can measure the animal's false alarm rate. And here, this is a much more moderate decrease in false alarm rate, but nonetheless is significant. So here's data across all of the experiments that we did across all of these different areas. And again, we see that when we suppress V1, LM, and AL, we see this robust increase in the animal's threshold for detecting these changes in contrast, but no effect when we suppress PM. Interestingly, we saw something slightly different when we were looking at the change in false alarm rate. Again, when we suppress V1, we see some decrease in the animal's false alarm rate, although it's less robust than in the orientation discrimination task. Suppression of LM and AL have no effect on the animal's false alarm rate, and suppression of PM actually increases the false alarm rate. And so this was really surprising to us, that this process where we actually decrease the activity of an area makes the animal more likely to think that there was a change that had occurred. So again, to investigate which of these different effects might be sensory versus cognitive, we move the visual stimulus outside of the area that we're suppressing. And now you can see that all of these effects on threshold go away. The animal no longer is impaired on the hit rate. And indeed, also the decrease in the animal's false alarm rate also goes away. But this increase in false alarm rate that we saw in PM is actually maintained, suggesting that while V1, LM, and AL contribute to sensory processing, that PM might contribute to a more global cognitive state in this contrast detection task. So in conclusion, what I've shown is that V1, LM, and AL, these three visual areas, are each required for the perception of both orientation and contrast. And that there's, I think, potentially a few different proposed circuits that could underlie this. Here, I've drawn out both the parallel organization that might occur where V1 might excite both LM and AL, which each converge on the decision-making target, as well as a serial option where V1 excites LM, which we know also excites AL and feeds forward to the decision-making area. And so this is something we're really interested in understanding, is what the circuitry is between V1 and the decision-making areas. We're also really interested in trying to understand what the unique roles for V1, LM, and AL are, and if there are any tasks that LM is responsible for and AL is not, and vice versa. So far, our data, we're not really able to distinguish what the relative contributions of these areas to behavior is, and that's something we're really interested in. Secondly, we've shown that PM isn't required for perception of either contrast or orientation, despite encoding both of these features. However, what we have shown is that suppression of PM increases the false alarm rate in a way that we think might be cognitive. And we have a couple of different hypothesized circuits that could underlie this. So for instance, one possibility is that PM through some circuitry provides inhibition to the decision-making area. And by suppressing PM, we relieve this inhibition, thereby disinhibiting the decision-making circuit and making the animal think that it saw a change when there wasn't. Conversely, we could imagine PM feeds back into the sensory system and provides inhibition through this circuitry as well, which might have a similar effect. So I think potentially the most important question, the one that we're really interested in, is whether there might be a perceptual role for PM at all, what that task might be, what that behavioral condition would be, and how we might be able to figure that out. So I'd just like to conclude by acknowledging Meow Meow Jin. She was a graduate student in the lab, a superstar who did all of this behavioral work, as well as a bunch of physiology that I didn't show. I'd also like to recognize my wonderful lab, people that give me inspiration on a regular basis, and funding. And thanks to all of you.
Great, thank you, Lindsay. I miss Dr. Jin, she was great. Wonderful, wonderful work. So I want to start the Q&A session now, and I'd like to begin this by sending over to our keynote speaker, Dr. Berman, to see if she has any questions to start us off. Marlene?
Thanks. Firstly, thanks to everybody for those incredibly interesting talks. I learned so much in such a compressed amount of time. It was really great. Maybe I should start off with Jenny, because your talk was longest ago and I don't want my memory to fail so greatly. So you showed us all this amazing data from these mixture neurons, which is really extremely interesting, except for the plaid case. My recollection, though, is that when you showed us initially the distribution of the population responses under some percentage of conditions, there was almost as many mixture responses, like 50%, under 50% of the conditions, as was true for the intermediate responses, which is a bit like the average. And so I'm wondering why you choose to focus on the mixture, or how do you think about the relationship between the intermediate and the mixture?
That's a great question. Thanks, Marlene. Yes, so we have another whole analysis method that we deploy on the intermediates to try to ascertain whether or not there's fluctuating activity on those as well, but where it's happening at a faster time scale. So we don't capture it with a sort of 200 or 500 millisecond window. You can't get as much power with that kind of analysis. That becomes a really difficult problem. I would say that we have some good proof of principle from one of the data sets. Well, two of the data sets, depending on what time period you look at. The strongest evidence we've seen so far has been from Vindrik Frywald's data in one of the face patches. So there it looks like at least some of the neurons seem to be fluctuating, within a 400 millisecond period of time between coding one stimulus and coding the other. So that's really exciting, I think. And one of the things we have planned is to collect data with much longer stimulus presentations to give us more power for that kind of analysis.
Thanks.
Did you have any other questions, Marlene, to start? Feel free.
Actually, I do have a question for Roberto. And my question is as follows. So you showed us these 4 really interesting experiments and kind of built the story up incrementally. I'll just start with one. So in study 3, in which you are showing us the multivariate pattern in these different regions, in some regions, the correlations, I think the RSA, seems to be higher in, say, visual cortex. In another area, like more anteriorly, it looks like it's in the right hemisphere. So you had like 4 little pictures of coronal brain sections, and it seemed to me that it depending on where you were in the posterior to anterior trajectory, there was switching between the 2 hemispheres for the signal, but I could be wrong.
Yeah, actually, we didn't see a clear lateralizations. There were some more in the left. I mean, even the perirhinal cortex was bilateral. I put an arrow in the left just because that was the most noticeable, but there was also in the right. So we didn't find a clear lateralization, which perhaps we should have expected more for language. but we did not. So I guess in the left prefrontal, we found some more lateralized left prefrontal cortex for semantics. But I should say that, I'm just presenting a simplified version. There were many effects, and some that were kind of fit with the simple prediction. Okay, visual representations in visual areas predict perceptual memory, semantic representations in semantic regions, anterior semantic regions predict conceptual memory. But we also found, sometimes kind of visual representations were predicting kind of a conceptual memory in more anterior regions. So this is what we are still puzzled by, these interactions between the type of representation and the regions. So I think that's topic for other studies to understand.
Great. Just a quick follow-up question. Again, it's about hemispheric specialization, and it's because that's the topic of my talk. So it's like a one-trick pony, or as the expression goes, something like that. So in your first study, you did identify a lateralized region, and that is the left OTC in your barn experiment. And so even that seemed surprising to me, that you would get such lateralization. And I wonder whether you can comment on that.
Yeah, well, the first one is focused on ERS. So perhaps, this kind of, so it's a combination of the regions where representations were stored during encoding and reactivated during retrieval. So it's a different type of evidence than the one in which we just use encoding representations to predict performance. So perhaps there lies the key difference. So, but definitely we should, I think we didn't put enough attention to look at lateralization, even though it has also been close to my interest, the lateralization effects, as you know, from aging. And so, yeah, we should probably go back and look more carefully at what happens with lateralization.
Thanks.
Thank you.
Great. Before we get to the Q&A from the audience, I wanted to get in a question for Lindsay. Have you tried using your optogenetic methods to suppress one area like V1 while recording from the other areas in that whole network like LM and AL to try to sort out the causal links and circuits between these areas for visual processing? You kind of touched on that at the end as a future direction. What are your predictions there? Do you think it's feasible to do that sort of kind of bigger circuit analysis using your approach?
Yeah, absolutely. So short answer is no, we haven't done that. I would love to do those types of experiments. People have done the inverse. So one concern might be is that when you silence a higher visual area, that if it feeds back to V1 and provides excitation to V1, that removing that excitation is essentially equivalent to suppressing V1. And so maybe that could be a concern, is that that the higher visual areas aren't necessary per se, but when you remove their feedback to V1, that causes a concern. So other people have done those experiments and generally find relatively subtle effects of silencing higher visual areas on V1 activity. But yeah, I think it's a really open question about how much V1 is actually responsible for the activity in the higher visual areas. And there have been some higher visual areas identified, for instance, this area called POR, which when you silence V1, nothing happens. But there's, and so the idea is that it might receive its input from areas like the colliculus. But there's a lot less known about LM, AL, and PM and how much of their activity is actually being routed through V1. And I think those are really important questions.
Yeah, sure. So I'm going to move to the Q&A from the audience. There's a question from an anonymous attendee for Dr. Cabeza. The question is, retrieval of memory is typically associated with large-scale univariate activation, particularly in regions of the DMN. But in the ERS analysis, it seems that only the left temporal region shows encoding retrieval similarity, the ERS. Would this suggest that perhaps in other regions, retrieval might not entail the reinstatement of the original representation?
Oh, this is a very good question. So Yes, so this study had the limitations of power. I mean, other analyses that look at DRS were finding other regions, including in the angular gyros, which is part of the default network. But I should say that in general, the default network also represents a lot of processes that are involved in doing retrieval. So it's not only representational kind of effects. So that is also the difference. The representation effects are often more localized, and in particular, if you look at this type of subtraction that we did, is that is item minus set, in which we've only focused on what is identical versus the whole set, because sometimes, I mean, it's only kind of based on just the set ERS. But there is actually some interesting evidence that there seems to be a shift from visual areas play a greater role in storing representations during encoding, but parietal regions have a greater role in representing information during retrieval. This has been called kind of a encoding retrieval shift in representations, but there's still, I mean, there are only a few studies showing that. So, but perhaps, I mean, that is related, because the angular gyros, for example, is actually showing deactivation doing encoding in other default networks too. So there's also this interesting flip that still is not understood very well.
Great, thanks, Roberto. Among the panelists, do you have any questions for each other? I think Jenny might have had a question.
I have a question for Lindsay. Really interesting stuff. So I'm wondering about the tilt after effect. You probably know this illusion, but in case the rest of the audience doesn't, if you stare for a long time at one orientation, my memory is that then your perception of a fresh orientation is a little bit pushed away from the orientation that you were overexposed to. Is that the kind of thing that you think you might be able to look at in this paradigm? Do you think that there's like a consistent shift in the direction that the mice are perceiving after suppression of a particular subpopulation?
Yeah, so that was actually something that we were really interested in. And surprisingly, it seems like the mice have the opposite effect. So for instance, like, we present this rapid presentation of the distractor gratings. And so you would think then that the mouse might be therefore better able to distinguish an orientation that's closer to the distractor as a target after it has gone through this adaptation. The actual finding that Meow Meow had, though, was it's the opposite, that the animal is actually much less good at detecting changes in orientation after more adaptation. And so I don't know if you noticed in the presentation of the movie that I showed you, the stimuli were coming at relatively irregular intervals. And that's because we took advantage of those intervals to condition the animal's hit rate based on the adaptation state. And so what she found was that the more adaptation there was, the worse the animal is at detecting. And it's because the animals are using a suboptimal method of doing this task. Essentially, they're using an all positive computation, that they're just listening for increases in firing rate rather than actually computing what the orientation is and then asking if that's a different orientation. And so this might be a limitation of the mouse model, or it might actually be what I'm very interested to know if it's what humans and non-human primates might do if trained on a similar task as well. And I think that that's kind of an open question too, but thank you for asking.
Too much screen time for the mice.
Yeah.
Great. Any other questions among the panelists? Marlene had another question.
This is also a question for Lindsay, but maybe it's a naive question, so I'm hoping you can tell me. So at the very end, when you showed us that PM seems to be more involved in inhibition, either in the decision or in the feedback to V1. It got me wondering, because the inhibition is specifically in the false alarm case. And so somehow PM knows that there's nothing on the screen, but the response has been yes, as in the case of the false alarm. It's not like the no trials. It's not like the yes trials. And so PM seems to be extremely sophisticated in kind of monitoring the type of the information. And I'm wondering whether you think that this is indeed a capability of these neurons.
Yeah, so I don't know about how sophisticated it is, but I think that's a key observation that you make. I think one other important observation is that we only saw this increase in false alarm rate on the contrast detection task and not in the orientation discrimination task. And so this is purely speculation about why I think that might be. I think that there is something about the noise regime that the brain is working in when you're asking the animal to discriminate very high contrast salient stimuli from each other versus detecting a very low contrast stimulus from the background. And so I think that PM might be altering that noise threshold in some way that is really exquisitely sensitive to that condition. And so, whether this then is really what PM is good for, or just some influence that it has on the circuit in this particular context, I think is a very open question. But yeah, there's clearly something not straight signal detection theory happening here. Yeah.
Thanks. Allison, did you have a question?
Okay, thank you. Yeah, I have a question for Roberto. I'm really curious about the anterior temporal hyperdifferentiation that you described, but you focused on for semantic stimuli, but it was also there for visual, right?
Actually, that was not significant.
Oh, okay.
So that effect, yeah, and for the visual. So it was, approaching, yeah, but not significant. So
well, that would be consistent actually with my question about it, which is that there might be individual differences related to a visual and hearing loss that people, so because there's, hearing, that the sensory losses are associated with cognitive shifts.
yeah,
So,
aging is associated with visual deterioration. Sometimes this is not sufficiently emphasized because we have glasses, but even with glasses, there is a loss that, just from the crystalline lens is by 880 is only kind of getting like, I don't know, remember, I think it's 20% of the actual light. So it's actually losses in the optical nerve. There are losses in B1. So there is even if you are not at the level that you need to rely more on. So, and then whether this change, in terms of relying more on meaning, it happens, gradually, or is the shape that this recruitment has on whether, but given that happens for other functions, it's not just for vision. This generally emphasis of meaning can be seen all across cognitive functions in older adults. Having meaning actually attenuates age effects. So older adults are much better at remembering meaningful things than meaningless. So this is probably kind of a gradual process that, since our knowledge is spared, so we rely more on what we know, and so this is our interpretation that is related to that. A shift of emphasis probably happens with attention. So
I think I was interested in whether we could relate it on an individual basis to the status of people's.
Well, we found that, there were variations in the ATL recruitment, and those were kind of also correlated with the sensory. So that the greater the sensory, the greater the ATL. So that would support also the compensatory, and so that it seems that there could be, a compensatory response.
Thank you.
So turnabout is fair play. I wanted to see if Lindsay had a question for Jenny or anybody else.
Yeah, I do, in fact. So I found the result with the plaids really interesting, that, you see this very different modulation of responses in that condition. And I like your story that maybe it's because this is a more unified percept. But I was also kind of wondering how this might relate to the composition of the plaids, that typically the neuron might be selective for one of those stimuli, but not for both of them. And how that might relate, whether you expect to have the same kinds of multiplexing in response to a stimulus that's preferred versus not driving, or whether you need it to kind of be in that dynamic range and how that might work, if that makes sense.
Yeah, it kind of does. And I won't be able to necessarily answer that for the plaid per se. But so one thing to say about the plaid is, for those of you who are not total vision geeks watching this, if you present two gratings and they're going like this, if you set the contrast right, the grading, if they're the moving grading, it can sometimes appear to be moving this way and not, or sometimes it can appear to be sliding across. If in that kind of a sliding situation, I would expect to have mixtures appear and, you know, both of those be represented, but these were stationary. So that you wouldn't have either of those conditions to consider. Getting maybe more at your sort of larger point of like, what is it about the stimuli? How does it relate to the receptive field and how the neurons are tuned for one versus the other? Sean Willett, who graduated about a year or so ago from my lab, he did the kind of a fine-grained evaluation of multiplexing across a range of different frequency separations in the inferior colliculus data. And what he found in that was that these mixtures were more evident when the frequencies were reasonably closely spaced. So kind of in the domain of the neural representation where the problem is most significant, like that there really is overlap in the population of neurons that's activated by the two stimuli. That's where there seems to be this elbowing for, you know, who's getting represented in which neurons. So I think there's much to be done in terms of more systematically exploring the separation between stimuli and the consequences for which neurons are getting activated and what the neural activity patterns are.
I like your idea of using the transparent versus non-transparent plaids as a good control, though, that seemed like that would sort out the issue of receptive field domain versus unified percept domain. Cool.
Yeah. And going back to Marlene's question about the intermediates also disappeared in the plaid case.
Was that your question, Marlene, or did you have another follow-up question?
I had another follow-up question, very close to Lindsay's question here. And this has to do with the single percept, you know, the single unified percept, as in the plaid case. But of course, there are so many times in the visual world where we actually do need to represent sort of two things. Like, I don't know, I was trying to think of a good example. I'm not sure I have one, but, you know, maybe this will suffice. Like, imagine if there's a spoon, you need to represent the handle and sort of the cup part, because of course those could be a fork as well, like, you know, two different components. So this is a case where there's sort of two things, but you really do need to represent both of those. You cannot have the inferior colliculus just thinking it's one thing.
Yeah, yes. You're absolutely right. And I think one wants to, you know, maybe go back to the Gestalt grouping principles and sort of finely vary the tendency for something to bind together into a single object, and see what's happening with this kind of activity pattern.
Great.
And I want to, unless somebody has another question for me, I want to kick it back to Roberto to say, what did you mean in the chat about the reading glasses changing your working memory capacity? Because that could explain a lot in my life.
Well, I mean, actually there is quite a bit of evidence that the visual deficits are actually correlated with cognitive deficits in older adults. So definitely visual kind of in general sensory kind of deficits contribute to cognitive deficits in older adults. I mean, either because it's already degraded information that flows through the system, or because they are both, well, associated with aging. So there's an indirect correlation, but yes, I don't know if that's what you're referring to in terms of glasses, but yeah, actually they're showing that when the information is presented to older adults in even larger fonts, that reduces the age effects.
My apologies, it was Allison's comment, but she said Roberto, so I misattributed the.
Sorry. I just was wondering when to mention the class.
Your answer was good too. I just didn't want to cut Allison off.
Oh, no, I was just giving my anecdote right, about the difference that I noticed when I had to start restricting my visual field based on focus, right? So then it's the working memory load of having to like go from place to place.
Oh, yes. I've noticed that as well.
Yeah, and as someone who did not need glasses prior to getting old, if I noticed the difference. So anyway.
Great. Well, thank you all. I think we're going to wrap up this discussion.